now in this today's objective, we will continue for the third week of development and what happens from the different germ layers and what are the supporting membranes of the embryo. Okay, so our objectives are the third week of development with their relevant anomalies. We will discuss about the derivatives of germ layers, and we'll discuss about the mainly placenta and different placental fetal membranes. Let's discuss about the third week of development. Now, as I told you, for the second week of development, it is a week of twos. Means everything is in two layers of trophoblast, two layers of embryo disc, two cavities. When you come to the third week, everything is threes. So you can see that the third week of development forms three main structures. The three main structures are the primitive strix, the notochord, and neural tube. As you can see in this slide, this primitive strix is known as primary organizer of embryo. What does mean by primary organizer of embryo is if primitive strix do not develop, then further development do not happen. That means if primitive strix is absent, further development will not happen and the embryo will be aborted. It will be removed. No development. So that's why primitive strix is known as primary organizer of embryo. As you can see in the picture besides the text, that there is a strix-like form in the caudal part of the embryo, and this is the cells which is forming the primitive strix. If these cells appear and that group of cells forms a strix, then the further development takes place. So that is primitive strix, and its importance is it is primary organizer of embryo. Without that, no further development. The next structure which appears is notochord. Notochord is the one which appears after the primitive strix. So notochord is a next step of development which is responsible for formation of neural tube. If notochord do not form, the neural tube will not form. Actually, neural tube forms the nervous system. So if the notochord do not develop, neural tube will not develop. If neural tube will not develop, nervous system means the brain and spinal cord will not develop. Right? So ultimately, we need three important structures during third week of development. That is the primitive strix, which is a primary organizer. It is required to be first step of development to form further structures. The notochord, which is responsible for formation of neural tube, and neural tube itself, which forms the nervous system. So these are the three structures we develop during third week. We'll discuss in detail in successive slides how these structures changes and what anomalies can occur. Now next is three layers will form. As I am telling you, third week means everything is three. So third week has a three layers of embryonic disc, and that is ectoderm, endoderm, and mesoderm. During second week, there will be two layers, ectoderm and endoderm. Now during third week, what happens is the ectoderm, endoderm, and mesoderm. The third layer, mesoderm, is added during third week of development. And the three cavities will appear during the third week of development. What are these three cavities? Amniotic cavity, yolk sac, and chorionic cavity. During second week, what we discussed? Amniotic cavity and yolk sac was there. Now what happened? One more cavity is appeared. That is what is the chorionic cavity. So you have amniotic cavity, there is yolk sac, and the chorionic cavity. You will see in successive pictures how these cavities look like and where the embryo is hanging within these cavities. So let's discuss about the primitive strix in little detail and its relevant abnormality. So as you can see that in the caudal part of the embryonic disc, which is seen from the top, you can see that in caudal part there is a strix-like arrangement that is known as the primitive strix. And this particular primitive strix is the one from where the cells will migrate to form mesoderm. So primitive strix ultimately is responsible to form mesoderm in the middle and forms the notochord in from the center part going out between the ectoderm and endoderm, and in the central part it goes up, as you can see the arrow going up in the cranial part to form the notochord. So The primitive stick is the one which is responsible for process of gastrulation. Gastrulation means what? Formation of mesoderm. So the process of gastrulation is responsible for formation of mesoderm, and this gastrulation is the primitive streak. So primitive streak ultimately gives rise to this particular mesoderm also. And in the cranial part, which is going up towards the head end of the embryo, this will form the notochord. So primitive streak is one of the very important structure. It fixes the whole structure of the body and axis of the body even. So if the primitive stick is in the center, then you will one single body. But if the primitive stick splits in the upper part, okay, then what will happen? Abnormal development. If primitive stick do not appear, no development. But primitive stick appear, but abnormally split. Let's take this example. Here you can see these twins. When the primitive stick in the upper part splitted, the baby has two heads, single trunk and the limbs, as you can see in the first photo here. So this is the Siamese twins or the conjoined twins. It is because of the abnormal primitive stick. Because primitive stick splitted in the upper part, so that's why two heads are developing and rest of the part is single. But suppose primitive stick splits in the upper part also and lower part also. So what you find is two babies, but they are fused in the middle, because primitive stick is set single in the middle, but upper part and lower part it is splitted. So that's why you find that the baby has two heads and has four limbs down, and that's how this type of abnormalities appear. This is known as conjoined twins, and this is because of the abnormal primitive stick. If the primitive stick continues to remain, it should disappear. Once notochord forms, primitive stick disappears. But suppose primitive stick still continues to stay, then it will tumor, which is known as sacrococcygeal teratoma. This is because of the persistence of the primitive stick. Usually it should be surgically removed, and it can be done. Same way conjoined twins, some of the type of conjoined twins can be separated, some cannot be separated. So this is what is importance of knowing primitive stick, which is a primary organizer, and its congenital anomalies. You should remember that these anomalies are very important to know in practice because of the conjoined twins. Mentioned, the primitive stick will give rise to notochord. As you can see in this particular picture, the black color in the particular disc is the notochord. So above is ectoderm, below is the endoderm, and in between in the center clear is the black color notochord. So this notochord ultimately develops from primitive stick, and later on primitive stick disappears, notochord remains. Once notochord appears, it will change the ectoderm above the notochord. So whatever is the ectoderm, that is blue color in this particular picture, clear above the black color notochord, this particular picture, the blue color above the notochord will become neural tube, which we will see in the next slide. So that particular neural tube will form above the notochord from ectoderm. So this particular part will be known as neuroectoderm. So that part will form the brain and spinal cord. But notochord, what will happen? Once the notochord gives rise to the neural tube above it, thereafter notochord is not required. So here notochord develops and then disappears. In the human body, it remains as just one ligament into the and some of the part of the intervertebral disc in the vertebral column. So we are the vertebrates, not the chordates, human. So that's why human has a vertebral column. Some animals have the notochord as a vertebral column like thing. So we are vertebrates. That's why notochord is not required in us. So notochord appears to give rise to neural tube and then disappears. Is it clear? So you should remember that in stages of development, some structures appear, they do their job, and they leave. Same way here, notochord appears, gives rise to the neural tube, and then notochord disappears. So what happened now? Next step will be the neural tube. Whatever is the part above the notochord in this picture, you can see the dark blue color given. This particular part is the neuroectoderm. Neuroectoderm is nothing but a part of the ectoderm just above the notochord. So this part changes and ultimately starts folding upon itself and forms a tube. This particular tube is known as neural tube. The margin of the particular fold is neural crest. So neural crest and tube are two components of neuratoderm. Neural crest gives rise to the adrenal gland medulla and some of the part swan cells and different parts into the body, apart from the neural tube giving rise to the complete nervous system. Neural tube is important for us to know at present as it will develop the brain and spinal cord. So we will go into the most important aspect of this is the neural tube. Neural crest cell gives rise to diverse structures in the body and they disperse into the body. Like they come from the brain and goes to the adrenal gland into the abdomen, and that is adrenal medulla. So it is coming from the neural crest cells. But our basic understanding for currently, at least we can focus that the neural tube gives rise to the brain and the spinal cord. Right. So what we discussed, three important structures. The primitive strict, important as primary organizer, and if it doesn't develop properly, it can lead to congenital anomalies like conjoined twins. Then we discussed notochord. It is important as inducer for neurotube formation. If it doesn't appear, the brain will not form, and neural tube itself, which is giving rise to the brain and spinal cord, that is nervous system. So we discussed that there were three important structures. This is the picture which gives rise to gives you idea about the three cavities, because we are going to discuss the layers in a little separate slides and their derivatives. Let's conclude this part about the cavities. As you can see in this slide, there are three cavities. You can see in the big cavity, which is chorionic cavity, and within this big cavity you see that there is a floating clear cavities, which is a small cavity above and a little bigger cavity below. Upper cavity is known as above the embryonic disc. Whatever is above it, we call it amniotic cavity. Whatever is below the embryonic disc is known as yolk sac. So you can see in this particular slide that it is like a two balloons floating into one large balloon. So two balloons floating into the large balloon. Two balloons are one above amniotic cavity, the other one little bit bigger below. It's known as yolk sac, and these two are floating into a very large cavity known as chorionic cavity. And this particular amniotic cavity with a yolk sac and embryonic disc is hanging into the chorionic cavity by a connecting stalk, which is going to form umbilical cord later on. So this is what is about the three cavities appear during third week of development. What are the names of three cavities? Amniotic cavity, yolk sac, and chorionic cavity. Later in the development, what happens is that amniotic cavity starts becoming bigger and bigger. It is the smallest cavity at present in this stage, but later on this becomes bigger and bigger, and then the yolk sac will go into the body of embryo to form digestive system, which we'll discuss when you will come to the College of Medicine in the first semester of the first year. So that is going to form the GIT, that is yolk sac. Amniotic cavity becomes bigger and chorionic cavity disappears. So ultimately, the whole body of the fetus will be floating into the amniotic cavity. At present, amniotic cavity is the smallest of all. But in the last stage of development, finally, amniotic cavity becomes the biggest, yolk sac goes into the body of embryo, and chorionic cavity disappears. Ultimately, it is obliterated. So this is what is about the three cavities. We discussed three structures, we discussed three cavities. Let's understand some of the anomalies of the nervous system, and then we'll go to the layers. As you can see in this particular slide, it is some of the anomalies of nervous system. As I mentioned, that the neural tube will give rise to the nervous system, the brain and spinal cord. What will happen that it is covered by the vertebral column. In some of the developmental errors, the vertebral column is not complete on the back, and that's why the skin is the only covering for the spinal cord. This type of condition is known as spina bifida occulta, because the spine is splitted, and that's why the vertebral column is not formed on the back, and the part of the spinal cord is just covered by the skin. I will show you the next slide, the photo of actual the patient, how it looks like. So this spina bifida occulta. When the meninges comes out from that gap in the vertebral column, when the meninges comes out, we will call it meningocele. What is meninges? Covering of the spinal cord. So covering of spinal cord comes out, but spinal cord still remains in vertebral column, then it is known as meningocele. If the spinal cord also comes out along with the meninges, we will call it meningomyelocele. If the spinal cord is directly open on surface, that is the worst type of congenital anomaly, which is known as myelocele. So we have spina bifida occulta to meningocele to meningomyelocele and myelocele. So these are the different congenital anomalies, which is because of the development of a developmental error of the spines of the vertebral column. Let's see the photo of this patient, how it will look like. Just to give you idea, I don't expect too much of this congenital anomalies except some basic which I am showing you now. This is what is the, you can see this baby has the black color hair in the back, and this is the area where the spinal cord is just covered by the skin. This condition is known as spina bifida occulta. While the this one is the meningocele, and here the myelocele where the surface spinal cord is open. If the complete nervous system will not develop because of absence of the notochord, then absence of the part of the brain and the skull, this condition which you see in the lower part of the slide is anancephaly. So anancephaly is nothing but absence of the brain and the skull. So this part is, this baby will not survive, it will be dead, it will be stillborn. So once the baby is born, it will be dead. It is usually detected by the blood sample from the mother, and alpha fetoprotein is the protein which we can assess to find out such type of anomalies. Any anomalies of nervous system can be very well and easily detected by the estimating level of the alpha fetoprotein into the maternal blood. So you take the mother's blood, you test it for the alpha fetoprotein. If the level is high, it suggests that something wrong in the development of nervous system of the baby in the uterus. So this is what is some of the clinical importance which I expect at least as a preparatory student to know, that we need to know what is this development, and if the something go wrong, what can happen. This to give you overview of such type of thing. As you can see here, now we are coming to the layers, right? So we are in the 3 germ layers which we discussed: ectoderm, endoderm and mesoderm. I don't expect that you can remember all the different names which can be there. I just put up some important facts in this slide, and I expect all the names to be remembered for this particular slide, because it is very basic, very simple, and important items I kept for you of the derivatives of germ layers. So let's understand from ectoderm what forms. Okay, nervous system, central nervous system, peripheral nervous system, and epidermis of the skin, and the hair and the nail. So these are the structures which comes out from the ectoderm. So ectoderm gives rise to the central nervous system, peripheral nervous system, epidermis of the skin, which includes the hair and the nail also. So these are the derivatives of ectoderm or formed from the ectoderm. What are the derivatives of the mesoderm? Mesoderm gives rise to the vertebral column, muscles of the body, and blood vessels including heart. So heart and the blood vessels develops from mesoderm. Vertebral column develops from mesoderm. Muscles of the body develop from mesoderm, usually the back, limb, body wall, etc. So this is what is the some of the important structures developing from mesoderm. And the ectoderm gives rise to the epithelial lining of different system, like gastrointestinal system or gastrointestinal tract, respiratory tract, or urinary bladder and urethra. So the lining of this particular tracts ultimately comes from endoderm. So epithelial lining of GIT, respiratory system, and urinary bladder and urethra, these are the parts which develop from the endoderm. So let's just understand, suppose we try to understand how we can remember these things. So let's play the game that what comes from mesoderm? Muscles of the body, vertebral column, and heart and the blood vessels. Nervous system develops from what? Ectoderm, mesoderm, or endoderm? Ectoderm. So epithelial lining of GIT develops from ectoderm, mesoderm, or endoderm? Endoderm. So you understand that whether you should know which system develops from which germ layer and which germ layer gives rise to which different system. So this will be the easy aspect, and some of the important names are only just one or three of the important structures I kept out of so many structures coming from these different germ layers. So this is what is about the three germ layers and what it gives rise to. Now, up till now, what we discuss is three important structures: primitive strict, notochord, and neural tube. What it gives rise to and what are their importance. We discuss three cavities: amniotic cavity, yolk sac, and chorionic cavity. We discuss three germ layers: ectoderm, endoderm, and mesoderm, and what it gives rise to. Up till now, we were discussing that embryo is like a disc, it's like a plate. But later on, as you can see here in this particular slide, that it starts folding, and there is a head fold and the tail fold. So head and tail fold comes together, and there is a lateral fold coming from the side. So ultimately, head fold, tail fold, and the lateral folds together ultimately fuse at the level of the umbilicus. And from there is an umbilical cord which hangs whole of the fetus clear from the placenta. So the plate-like embryo becomes a cylinder-like embryo. So human shape is changing from disc to a cylinder, and then it gives rise to the human shape. So as you can see, this is the lateral folds, and next slide can give you idea about how the cylinders looks like in the embryonic development. So here you can see that this particular shape has changed just like a simple cylinder to partly human-like shapes, but still its small livers are seen. You can see that's umbilical cord. You can see a head with so many grooves and the elevations. So the heart bulge is seen in the center. So you can see that it still doesn't look like a human embryo, but shaped like a human. Gradually changes to the contour of the human. As you can see in this particular picture, that somewhat it looks like now the human baby or newborn baby like thing. So gradually our embryo development changes from the plate-like embryonic disc to the cylinder and then the human appearance. So at this point we will conclude a part as far as the third week of development and their overview is concerned to form different systems. Later on, different systems form different part. Then cardiovascular system, GI, digestive system, respiratory system, nervous system, and how they function, urinary system, how they function, how they start changing their different structures. This will be the detailed systemic embryology which you will learn when you will come back to the College of Medicine. And during the different blocks and relevant structures, you will learn the development of those structures there. So this is about the development of the different parts during third week. Now we should just go through different fetal membranes or supporting structures. The supporting structures are, the most important is the placenta, because the developing baby or the fetus will need a nutrition. What happens is the ovum is the one large cell and sperm is a cell meeting to the ovum and forms a zygote. So the cytoplasm of the large size ovum ultimately supplies nutrition in the initial stage of development. Later on, it implants into the fundus of uterus, which I told you in the beginning when we started this presentation. That implantation means the ultimately the blastocysts implant into the fundus of uterus, and the tropoblasts surrounding blastocysts fundus of the uterus, and this part will form the placenta, and from there the nutrition for the developing fetus will come. So the placenta is a structure from by which ultimately the developing fetus receives nutrition. It will be what? Exchange of the maternal and the fetal gases and the nutrients. But do remember that at the placenta there is no exchange of the blood. Mother's blood and the fetus blood is totally separated by a membrane known as placental membrane, and from the mother side there is decidua, which is glands and the blood vessels of the uterus. So that is, decidua is the maternal side of the placenta, and the finger like villi which is formed known as chorionic villi. So chorionic villi is the fetal side of the placenta. So fetal side of the placenta is formed by chorionic villi. It is a part of chorion which we discussed for the chorionic cavity, and decidua is the nothing but the endometrium of the uterus. It's a lining of the uterus which changed its structure to give nutrition to the developing fetus. So these are the two components of placenta, and placenta is a disc shape, okay, having 15 to 20 centimeter diameter, 2.5 centimeter thickness, is usually around 500 grams. So this is the actual some of the facts regarding the placenta. But its most important fact is that it is responsible for exchange of gases and the nutrients between the mother and the fetus. But the blood between the mother and fetus do not mix. They are separated by placental membrane. So this is what is about the placenta. Other functions of the placenta apart from exchange of gases, so oxygen will come from mother to the fetus, carbon dioxide will be released from the fetus to the mother, and it will be then circulated back to the mother's blood. The nutrients, glucose and etc. comes here. Waste products like urea etc. goes back. So ultimately it is the exchange site, placenta. Apart from it, it is a defense barrier. So some of the viruses and bacteria cannot cross the barrier. So that's why baby will be protected. But there are some bacteria or viruses or some drugs which crosses the placenta. If they cross the placenta, they will be affected and baby will have a problem. For example, some of the rubella virus, clear? So they cross it and they can cause the damage to the development of the baby. So nature created a mechanism of defense barrier, but some overcome this barrier and goes to the baby. Some drugs can cross the barrier and that can affect. That's why during development or early stages of the development, it is not advisable except necessary, very important drug, and it should be safe from during the pregnancy. If it is tested that it is safe during the pregnancy, then it should be given. Any drug and every drug cannot be taken during the pregnant state, because the placenta is the barrier and sometimes the barrier is broken by these drugs and drugs will cross it, and then it can cause damage to the developing fetus. Apart from this working as a defense barrier, the placenta is also responsible for production of hormones. What are these hormones? The human chorionic gonadotropin, which is known as HCG. Also known, another hormone is a human placental lactogen, HPL. So these are some of the hormones which are formed from the placenta, and their job is to maintain the pregnancy. The human chorionic gonadotropin hormone, HCG, is responsible for maintaining pregnant state. If the level of the HCG is low, then the development cannot continue further and the baby will be aborted out. So the female who has repeated abortions, we should start thinking and assess the level of the human chorionic gonadotropin hormone. If the hormone level is low, you can give injections of this hormone and can maintain the pregnant state. So this is very important to know this hormone and this level, and then in practice we can apply this principle. So placenta is what? Ultimately the structure for the exchange between fetus and mother, but the blood do not mix. The fetal side is the chorionic villi and maternal side is known as decidua. The function is exchange of gases and nutrients, defense, and hormone production. What is the hormone? Most hormone is human gonadotropin, HCG. So this is what is about the placenta. Let's talk of some other membranes in very superficial, not go into much detail. We'll just have a superficial overview of different membranes. So other membranes other than the placenta, which is the one of the most important structure during development, other membranes are the chorion, amnion, yolk sac, allantois, and umbilical cord. So these are the other fetal membranes. Apart from placenta, we discussed. Now these are, we will take one slide each and 2 lines in each about this particular membranes. So let's talk of the chorion. What is chorion? Ultimately, it is formed by the trophoblast. What was the trophoblast? I told you at the very beginning that trophoblast is nothing but the outer shell of the blastocyst, which is a covering of the blastocyst. So this trophoblast ultimately gives rise to chorion. Chorion will ultimately form the finger-like projections, as you can see in this slide. This is known as chorionic villi, and this chorionic villi ultimately will form whole placenta, and this villi will float into the maternal blood of decidua other side. So blood ultimately touches this particular villi, but within the villi is the fetal blood. So fetal blood and maternal blood do not meet because these villi separate them, and that's why the oxygen and gases will exchange at this level, and then the mother's blood will return back and fetus blood will return to the fetus. So the chorion is important, which ultimately is going to give rise to the chorionic villi of the placenta. So when I talk of placenta, I told you that it contains chorionic villi, which is the fetal side. The chorionic villi comes from the chorion, and chorion comes from trophoblast. So this is what is importance of knowing the chorion. Chorion is one of the membrane, as I told you, and within this is a cavity, which is chorionic cavity, which we show in the 3rd week of development. Let's talk of 1 or 2 lines for the amnion. Amnion is the ultimately membrane which lines the amniotic cavity, and amniotic cavity contains a fluid. What is the level of the fluid? 500 to the 1000 ml. So 500 to 1000 ml is the normal quantity of the amniotic fluid. If the quantity of amniotic fluid is more, if it is more than 2000 ml, then it is known as polyhydramnios. Poly means large. So quantity of the fluid is more. It gives idea to us that there is an abnormal digestive system, or there is an abnormal development of nervous system, central nervous system. If there is an abnormal central nervous system or abnormal digestive system, it gives rise to polyhydramnios. So the fluid is more than 2000 liters. 2000 ml. So 2 liters blood, amniotic fluid. So this particular, if the quantity of the fluid is more than 2 liters, then it will be polyhydramnios. If the fluid is less than 500 ml, then it is known as oligohydramnios, and it is suggestive of abnormal urinary system. So abnormal urinary system gives rise to oligohydramnios. The amniotic fluid will be less than 500 ml. If there is abnormal development of digestive system or the central nervous system, the quantity of the fluid will be more than 2000 ml or 2 liters. If it's more than 2 liters, it will be known as polyhydramnios. So measuring the quantity of the fluid into the amniotic cavity is also clinically important to know whether the developing baby has abnormal development of either digestive system, nervous system, or urinary system. Fluid more, development error into the digestive and CNS. If the fluid is less, abnormal urinary system. And that is what is importance of knowing the amniotic cavity and amnion. As you can see in this slide, that the 2 balloons floating into one large balloon, amniotic cavity, yolk sac, and chorionic cavity, Gradually, as you can see, the size of the amniotic cavity is becoming bigger and bigger. Yolk sac is becoming smaller. The chorionic cavity gradually becomes smaller. And at the end, what you see at the lower part of the picture, that whatever whole cavity you see is nothing but amniotic cavity. So amniotic cavity becomes the largest cavity at the end during development. The next is the yolk sac. Yolk sac is the cavity below the endoderm. Above the endoderm, amniotic cavity. Below the endoderm, yolk sac. And this yolk sac ultimately forms primitive blood cells, primordial germ cells, which is for the sperm and the ovum, and it forms part of the GIT. So importance of yolk sac is to know that it forms the blood cells, primitive blood cells, and it forms a primitive germ cells. Germ cells means sperm and ovum. So this sperm and ovum starts developing into yolk sac, and later on it goes to the testis and ovary. So this is what is importance of knowing yolk sac, and it gives rise to the part of the gastrointestinal system or GIT. So this is what is about the yolk sac. Allantois in human is a small pocket, and it is rudimentary. So just sake of mentioning, I am mentioning that it is one of the membrane, but it is rudimentary in human, and it's a small pocket. The umbilical cord is a cord-like cylindrical structure, and it is covered by the amniotic membrane. It connects the fetus to the placenta. As you can see in this particular photo, the fetus is suspended from the placenta by connection of this particular umbilical cord. So umbilical cord is covered by amniotic membrane, and it contains, most importantly, we should know that it contains 2 umbilical arteries and 1 umbilical vein. So there is artery and the vein, but how many arteries? 2. What? They are known as umbilical arteries. And what is the vein? Umbilical vein. So there is 1 umbilical vein and 2 arteries into the umbilical cord. Arteries carries a deoxygenated blood from the fetus to the placenta for oxygenation, and the vein carries oxygenated blood from placenta to the fetus. So in this particular blood vessels, the circulation is reversed. So circulation is reversed like the pulmonary circulation in the body. Here is the circulation reverse. So fetal circulation is a reverse circulation. So the umbilical arteries carries deoxygenated blood, and umbilical vein carries oxygenated blood. So this was a reverse circulation, you should remember, during in the umbilical. Apart from it, umbilical cord contains yolk sac, allantois, and some connective tissue. But at least we should remember the most important content of the umbilical cord is umbilical arteries and the vein. Arteries are 2 and vein is 1. The length of umbilical cord is around 40 to 60 centimeters. This one is the length. If the length is more, it is also problematic. If length is short, it is also problematic. So we should know that if the length is more than 80 centimeters, it is abnormal. And what will happen? Because the length is more and the fetus is floating into the amniotic cavity, the umbilical cord can wrap around the neck of the fetus, and then it can strangle the neck of the fetus. And when the baby is being delivered, it may strangulate the neck, and maybe we will be died. So this is what is very important to know. Actually, what is the length of the umbilical cord. If it is short, it will cause problem, because during the delivery, if the baby is going out and the cord is short, it may torn and leads to bleeding. So either the length is less or length is more, both the situation is a problem. So we should know that the length of umbilical cord should be optimal, and it is around 40 to 60 centimeters. If the more than 80, it is abnormal. Less than 35 is considered as abnormal, and both has a problem. So this is what is about the umbilical cord. So we discussed about some of the fetal membranes after the placenta. Placenta we discussed as important structure for exchange of gases, nutrition, defense, and it has 2 parts, decidua and the chorionic villi. Then we discuss about the chorion, we discuss about the yolk sac, we discuss about the amnion, amniotic cavity, we discuss about the allantois is one line, not significant into human development, umbilical cord and its importance of its length, and the umbilical arteries at the vein and their circulation. So this is what is about the some of the important aspects of the development and some of the supporting structures during the development. As you can see in this photo, what happens as the full-term baby looks like this into the uterus, as you can see, and non-pregnant uterus is a very small size, and the pregnant uterus is quite enlarged and expandable. So there's tremendous flexibility about these particular organs to have the adjustment for the pregnant state of the female. So all these things are important for us to know, that we can assess these things by the technology now is available, sonography and analysis, like you can take blood analysis for the mother, you can take the amniotic fluid and analysis for amniotic fluid, even you can take a part of the chorion and analysis for the chorionic biopsy. You can do a sonography, which is non-invasive procedure. You don't have to do anything except to put a probe on abdomen, and you can see on the screen the size of the baby and the size of the head of the baby, any abnormalities developing into it, the brains and etc. So antenatal care to the analysis of whether the development is progressing normally or not is very important, because we can notice all these things early and we can inform the couple in advance what is going on in the development of the baby in uterus. So this is what is just a superficial, very simple and easy version of the development